Okay, guys, we're going to get started. It's a pleasure to introduce our next speaker, Dr. Maria Walker. She's a clinician scientist at the University of Houston College of Optometry with the lab focus on keratoconus and ocular surface disease research. She graduated with an OD and MS degree from the New England College of Optometry in 2013 and completed a one-year cornea and contact lens residency at Pacific University in Oregon. Just last year, in April 2021, she completed her PhD in physiological optics at University of Houston with a dissertation entitled The Impact of a Scleral Lens on the Eye, in part evaluating the inflammatory cytokines in ocular surface tear fluid using Luminex assays. In September 2021, Maria started a position as a tenure-track assistant professor at University of Houston. She is a clinical attending in the cornea and contact lens service at the University Eye Institute 2 mornings per week and spends the remainder of her time on translational research projects, including evaluating ocular surface immunity in keratoconus. The title of her talk is Scleral Lens Wear, Measuring Inflammation in the Fluid Reservoir. So Maria, I'd like to welcome you.
Thank you. Okay, thank you very much, and it's a pleasure to be here. So my background is a lot different than our two speakers this morning, and I also am quite a bit earlier in my career than them, like was mentioned. I just finished my PhD last year. So part of what I'm going to talk about today is some of the data that went into my dissertation. But first, just a general kind of overview to add to what Bobby said. You know, I am at the University of Houston, so we do have a full-service eye clinic there, so I do see patients a couple mornings a week, but primarily I'm in the research lab. And so our lab, actually, Carolina Lima is my lab manager here, and Justina Assad, my first PhD student, is also here. So we're very excited. I learned a lot from you guys this morning, and I'm going to share with the rest of you kind of the experience of doing ocular surface research, because I don't think that there's anybody else in here that really works in this space, and talk a little bit about some of the inflammatory diseases that we're studying. So just as a quick overview, we'll talk a little bit about just ocular surface and ocular research in general, because that's a relatively foreign probably to this audience, and talk about measuring or looking specifically at the tear fluid, which many people have probably never even thought about unless maybe you have dry eyes or you wear contact lenses. And we'll talk about how we use the Luminex technology similarly to how we test for blood proteins. We do this in the tears. And then we'll talk a little bit about keratoconus, which is nobody knows how to pronounce that. Everybody's always keratoconus. So I'll tell you now, kerato is Greek for cornea. So you maybe have heard that term, front clear surface of the eye, and conus, conical. So it's a conical cornea, and we'll talk a little bit more about that towards the end. So where do we start, right? Here's the schematic of an eye. We're looking at a side view. So you've got your retina back here, macula. All this is very important. The optic nerve going back to the back of the eye. But any of us that do research know how niche everything gets. So I don't know very much about this part of the eye anymore. It's been a long time since I took boards. And so where I focus is this front part of the eye, and here's our cornea there. And so that's really where the focus of my research is. Here's just another schematic of the eye. So you're missing that whole back retina, macula, all that nerves, important stuff. We focus here, front of the eye. So this area in general is called the anterior segment. So this is going to consist of the iris, which is the colored part that makes your pupil big and small, and then the lens behind that. So these, between the lens and the cornea, you're going to have all the focusing power of the eye. So of course the eye is an optical system. It works similarly to a camera. You have these lenses that condense the light. So the cornea actually contributes to about 60% of the eye's power. So it's an optical device, but it's very susceptible to the external environment and inflammation, both in the tissue itself, but in the tear film, and we'll talk about some of the diseases associated with that. This entire chamber is filled with fluid, which is called the aqueous humor, and then we have, again, this cornea. You've got the conjunctival tissue, which anybody who has had conjunctivitis, right, inflammation of that conjunctival tissue, that will get pretty red and inflamed. The cornea, if you've ever had a keratitis, inflammation of the cornea, usually comes with quite a good deal of pain associated. You don't typically forget when something like that occurs. And then, of course, we have that tear film. And the tear film has, incorrectly, I will say, but it has been called the extracellular matrix of the corneal epithelium. It's missing a lot of the ECM proteins and features, but the point is that the corneal epithelium gets a lot of its nutrition and can be really strongly impacted by what's going on in the tear fluid itself. And so looking at the most general schematic of the tear fluid, you can see this very first pink part is your corneal tissue, and then you have expanded out different layers of the cornea. Now this is a somewhat archaic model because it separates the mucin layer, most superficial or most proximal to the cornea, with the aqueous or primarily water-based layer and the lipid layer here at the surface. We know that this is much more of a mixed model, and so you can see this is probably one of the more famous schematics that's drawn, and so you can see here is microscopy of the corneal epithelium, and you've got these microvilli here, and on those microvilli you have a glycocalyx, which is glycoproteins that help to tether the main tear fluid to the corneal epithelium. So you can see a lot of interaction between that corneal epithelial surface, glycocalyx, and then all this fluid in here, which is the tear fluid. Now, specifically, what is in here and how thick is this fluid? So it's only about 3 to 5 microns in thickness. My audience is usually clinical, and I usually have to explain to them how much that is, but this is an audience where I do not have to explain what a micron is. So it's a very thin film. The lipid layer on top is on the order of 30 to 40 nanometers, so really quite thin. And this entire tear film volume is only about 20 microliters on the entire ocular surface. And every time you blink, a lot of that is turned over. Now, in the actual fluid, we've got thousands of proteins, lipids, metabolites. The lipids are primarily coming from the glands. You have glands in the eyelids called the meibomian glands. Those are secreting, most of those lipids are being secreted straight from the gland itself. You also have some blood that's being filtered through the gland, so you're getting, in some degrees, you're getting a blood filtrate in this, in which a lot of researchers have tried to monopolize on. You may have heard of glucose sensing contact lenses and other types of contact lenses to sense certain metabolites or proteins in the tear film in diagnostic purposes. Not so straightforward, but ultimately the tear fluid is coming in part, some of it is coming from the blood. So the lacrimal gland is going to produce most of that aqueous water-based fluid containing a lot of proteins. Most of these proteins are antimicrobial proteins, lactoferrin, lysozyme. And there's also what we focus on, a lot of inflammatory cytokines and proteases that are present in the tear film, and a lot of different diseases where we see irregularities in them. So we also have some production from corneal epithelial cells, conjunctival epithelial cells. We have goblet cells that will produce mucins, and those are embedded into the conjunctiva. So a lot of ocular surface cells will produce some of this fluid, but primarily you're talking about lacrimal gland and the meibomian glands primarily creating the tear film itself. Here's just an interesting image, and this is actually not to do with my particular research. I just showed this almost as an aside for those of you who are still kind of wrapping your head around the tear film and how we actually approach it and look at it. This is just a simple interferometry measurement of lipid layer thickness on the tear film. And so The light that's being used will interact with this is the cornea. You can see that patient's eyelash is here, and that tear film overlying kind of has an iridescent quality to it. And you can see if you take away the still image, you get the reflection. And basically, the more reflection or the more bright that these images are, the thicker that tear film is. And a reduced lipid layer is associated with all kinds of different diseases of the ocular surface, most of which result in some sort of pathologic dryness of the tissue. And dry eye is one of those things that you don't really think that much about unless you have it or know somebody else that has this condition, but it affects over 50 million people in the United States. So and some of this is related to autoimmune diseases and other systemic conditions. Some of it is environmental, and a lot of it is of course, both and combination factors. So in terms of ocular surface research using Luminex platform, we are not the only lab. So the Redfern lab, which is what has the MagPix, which is what I used in my PhD research. So this is from Rachel Redfern. She's also, she was my PhD advisor at the University of Houston. And essentially what we do is we use these instruments the same way it would be used for blood plasma, but we use our tear samples instead. And of course, there are some limitations. I'm going to come back to that in a second. Some limitations for doing this, right? So the Luminex platform's designed for plasma, and we're very used to talking to talking to some of the researchers who help us troubleshoot, and we're very used to saying, yes, we know it's calibrated for use with plasma. We use them with the tears. So you are limited typically mostly in volume, but also on normalizing the data, because we don't have, the equivalent of a regulatory gene that's this equivalent in all tear samples. So we have some ways around that, but it is certainly a limitation. So like I said, the volume of tears on the ocular surface at any given moment is going to be around 20 microliters. So when you're collecting these samples, it's very difficult to get a significant sample. And we'll talk about what the title of the talk, which is the Scleral lens, which helps us get a little bit larger sample of these. But typically with regular tear volumes, you're going to have between 10 up to about 80 microliters that you can collect. But that is on the very high range. Usually it's less than 20 microliters of fluid that you're going to get. So we either have to pool the samples between groups, which I don't like doing because we see quite a bit of variability from patient to patient, or we can dilute the samples with PBS, which can affect our CV values and just the efficacy of the measurements that we get. So we do have some limitations. What we do to normalize the data is we actually will load 10 micrograms of total protein. So we'll use a spectrometer to measure our total protein content in our tear samples. And then we load 10 micrograms of total protein for each sample. So then we normalize the concentration of usually inflammatory cytokines to in usually picograms per microgram of total protein. So if anybody's got any suggestions or better ways to approach that, we're always interested, of course, in that. And as I mentioned, I am a young investigator, so my use of the instrument is still pretty limited. So again, this is a fantastic, if I haven't said that, a really great event for all of us to get together, especially for those of us who maybe don't know everybody and as familiar with it. So looking kind of backstepping to the study that we actually were doing during my thesis. And so we talk about scleral lens, and again, everybody always asks me how to pronounce that word as well, because it's something that you're not used to hearing, and the scleral lens is, again, a word that we're not super familiar with. So those of us maybe who went to medical school might remember that there's something called the sclera, which is the collagenous tissue. The white part of the eye is that sclera, and that is why the lenses are named for that. They land on the sclera, so they actually vault over the entire cornea and land on that spongy conjunctival tissue. Again, we're looking at a schematic of the eye. You can probably tell something looks abnormal with that corneal tissue, and it is. These are not lenses that you or I would typically wear. It's more for a patient who has some sort of disease like keratoconus. But we are seeing more and more normal eyes, so to speak, being fit into these essentially prosthetic devices is how they're termed. If we look, this is an optic section, what we call an optic section. It's when you're looking in a biomicroscope, high resolution, at the front of the eye. So this is actually the cornea, this kind of gray tissue. You can sort of appreciate the edge of the pupil, so obviously a very high resolution. Here's our iris here. So here's the cornea, and you can see the lens, the front surface of the lens there is your very anterior-most reflection. This green area in between, that's the tear fluid. So you've got a tear fluid that normally on the ocular surface is maybe 3 to 5 microns in thickness, and now we've inflated it to about 250 microns in thickness. And so a huge part of my research and what I was focusing on during my dissertation was characterizing what this fluid is and establishing whether or not it has an inflammatory phenotype, and then ultimately determining, and these are our long-term goals, determining how we can study diseases from this fluid, but also how we can introduce therapeutics and manage these corneal diseases or ocular surface conditions using these prosthetic devices. So kind of a lot going on, a lot to do, but looking at, well, I'll share what we actually found looking in the fluid reservoir. Now, again, just mentioning that these lenses aren't going to be fitted on somebody who has, quote, a normal eye per se. So keratoconus is the most common condition. You may be able to appreciate here that this is not a very clear dome. So imagine you're looking at a lens, so to a camera or something, right? Should be very symmetrical. You should have an apex in the center. This person has an apex and inferiorly, so a lot of distortion. So I explain this sometimes to patients as saying it's like if somebody took a camera lens and like did blown glass stuff, right? Everything looks distorted. There's no real, it's really hard to define features and ultimately see. Here's a condition where somebody had surgery on their cornea. So all sorts of different conditions that can cause irregular optics, and we use these lenses for those. We also have a whole host of other conditions, and these are just, again, pictures of the eye. We put sodium fluorescein dye in so that you can appreciate what the tear film actually looks like on these patients. Probably don't have to be an eye specialist to know that that just looks like it's not very comfortable, right? So you should see this kind of smooth green interface if you have a smooth tear film, which neither of those patients have. So lots of different conditions. Hopefully none of you in this room have these conditions. If you do, come talk to me after. But you know, it's a real struggle for these patients to live and have productive, certainly visual lives. So these patients are fit into these lenses. So the actual technique that we use to collect this, you can see I have the patients look down, carefully remove the lens, and you can see this is just an image of a lens being removed from the eye. So they're really these large, rigid plastic prosthetic devices. But you can see you get a nice little fluid reservoir. So that's what I say when I say fluid reservoir, right? So this is the fluid that mixes with the tears and is trapped on the ocular surface. And just as an aside, we typically will use something like a sterile saline solution, a preservative-free solution, to actually fill the bowl of that lens, right? So that you don't have empty space and this dryness. So the start is saline, which ultimately mixes with the ocular surface tears. And so we have two types of tears that we collect here. So and I think I forgot to go back to the initial slide, but that's okay because we see it here. So one of the ways that you would normally collect the tears, so this is without a scleral lens, this is the most common way to collect the tears to study them, is to use a small glass microcapillary tube. And these work on the principle of capillary action, right? So the tubes will actually wick tears into the little vials. It's usually you can fit about 5 to 10 microliters into those little microcapillary vials. It can take upwards of 20, 30 minutes to really get a good sample from patients, and you have a lot of complications. The biggest one being you induce reflex tearing in this case a lot. You can imagine if you go and touch the eye here, you're going to induce some tearing, and those tears become watery and they can actually affect the composition of the tear fluid that we're looking at. But as of this point, it's kind of one of the better ways that we have. So what we did in this, and we just used normal soft contact lens wearers for this part of this study. This is very new type of research. I don't know of any other labs, maybe one other one in the world, our collaborator, that actually collects this stuff and look at it. So it's like we know nothing. So this is kind of the first study to learn anything about it. And so we did a complete anterior segment exam, of course. We fit with scleral lenses that are large diameter. It's all kind of relative. Anything over about 14 is considered large diameter lens. The subjects present to the clinic. We essentially measure the fluid before any lenses are introduced, and then after the introduction of lenses, we take those basal tears. So when I say basal tears, I mean the ones outside the lens margin, and we also look at those fluid reservoirs and compare the two. And so to do this, again, we're using the mag picks in Rachel's lab. We have since gotten the Luminex 200 for my own lab, but this was done using the mag picks. And we measure the total protein content, like I mentioned, using the direct detect, because that's our method of normalizing what we actually are getting for our results. And we also look, that's also why we take our baseline measurement, of course, as some control so that we know what the status is without a lens on. So the cytokine panel that we looked at for this particular project was relatively limited. We looked at a couple of the interleukins and a couple of the matrix metalloproteinases. So interleukin 4 and interleukin 8, both associated with inflammation, IL-4 being more specific to allergies. These are two cytokines that we tend to see increased when people wear soft contact lenses, so this is one that we wanted to look at. Also considering that we wanted to look at certain cytokines that could potentially be being produced by the corneal epithelial cells, thinking that they might get trapped in this fluid and be increased. We also looked at a couple MMPs. So MMP9 is the most ubiquitous MMP that's on the ocular surface. As an aside, it's also very associated with keratoconus development, which I mentioned is our disease of interest choice. MMP10, I think, one of you earlier was mentioning the, fight between, is it a good cytokine, is it a bad cytokine, is it maybe good sometimes, maybe bad sometimes, and I think we all can probably appreciate that sentiment. So MMP10 on the ocular surface, and I believe in most tissues, is typically plays a regulatory role, so actually dampens down inflammation. So we wanted to look at some that were pro-inflammatory and one that was more anti-inflammatory. We also look at simply ocular surface staining. So you can actually use sodium fluorescein, like I mentioned, to look at the ocular surface and see, and that's a way of looking at kind of functional stress of the tissue. And so again, we had our basal tears, we had our fluid reservoir tears. We measured them at the baseline and then at day 1 of lens wear, and had the subjects wear the lenses for 4 days daily, taking them off at night. And we measured after a few days to get some sort of more chronic, even if it's just a week or so of wear. And so if you look at the tear volumes that we were able to collect, so again, we've got baseline, and then here's day 1, from the basal tears, from the fluid reservoir tears, in day 4. So like I mentioned, you usually get, and you're going to get a little bit more fluid collection through the tear reservoir fluid than you would from just basal tears, but you can see not much more. We had some good collections on that last day. But total protein content, you see we get a little bit more, not significantly so, but you tend to get a bit of a trend towards more protein content building up in this fluid reservoir when compared to basal. So there's several different ways to look at it. For this, I just share I'll bring this up too. I have the median data here on the bottom. And then here is just following the individual subjects. And so you can see this third and fifth tab here, those are the fluid reservoir samples. And so you see often the trend is to see a little spike in the fluid reservoir with some of these cytokines. You can see here the only one that was significantly different was the interleukin-4 was greater in the fluid reservoir after 4 days of scleral lens wear. If we look at MMP7, 9, and 10, MMP7 really had no relationship at all between concentration in the fluid reservoir and on the basal tiers. MMP9 and MMP10 both showed, MMP10 most significantly did show that they were almost tenfold greater in the fluid reservoir when compared to what was on the basal ocular surface. And so to summarize the main findings of this, and there's a lot of data I didn't show, but we didn't, when comparing it to actual staining on the ocular surface, we didn't see what appeared to be a stressful situation, at least on the macro scale at the level of the corneal epithelium. But certainly we're starting to establish that the fluid reservoir is going to be distinct from the ocular surface basal tiers, and does probably harbor greater concentrations of some of these cytokines. Again, we need to look at larger panels, of course, because we have all sorts of different relationships between these molecules. We think that this is probably due to some limited tier exchange, which we know we're not getting a ton of exchange from the outer environment and the inner environment. And we think that these are probably mostly being secreted from the corneal epithelial cells. So this is a early pilot study. So a lot of it was established to develop the actual protocol, but also is leading us to different studies now to look at the effects of different therapeutics in the fluid reservoir in certain conditions, and also using it more specifically now looking in keratoconus as a larger capture of fluid that's more directly in contact with the cornea, which is where that disease is located. So our current lab projects, and I'll just talk for a couple of minutes more specifically about keratoconus. So we did just start up September of this year. We are looking at several different aspects of keratoconus, but the proteomics and gene expression, specifically in the context of immune cell infiltration onto the ocular surface, is really the direction that we're currently going. And I just will go through these quickly to show you clinically what this keratoconus, we also call it KC. You can probably appreciate this patient's right eye looks more conical or more protruded than his other eye. So it's typically going to happen in one eye, a lot more severe than the other. You get some iron deposition, some stress lines that can affect vision. I'll just go over this because I want to show you what really kind of is more meaningful to people who aren't clinically, which is what the world kind of looks like to these patients. So most of us have probably experienced some type of blur. Anyone who wears glasses has experienced blur on sort of what we call the first order. These are called higher order aberrations. So it's hard to correct this and really impossible to correct this without the use of some sort of rigid plastic contact lens, which is why these patients use lenses. It's associated, for those of you who work on other diseases, associated with lots of other conditions, specifically connective tissue diseases or collagen-based diseases. So Ehlers-Danlos, which is connective tissue where you have reduced proteoglycans, Lumican, Keratoconus, and Decrin, also present and also reduced in the cornea in keratoconus. Mitral valve prolapse, and just quickly here, if you look at the odds ratio of if you have keratoconus, your odds ratio of having mitral valve prolapse, and then the opposite here, odds ratio increases both if you have one condition or the other of having the opposite. And the other, which is probably one of the more interesting and certainly current studied, which is the association with sleep apnea. About a quarter of patients with keratoconus either have or go on to develop sleep apnea, and the odds ratio, depending on the study you look at, is pretty significant, and the prevalence in keratoconus patients is about 20 times higher than in the general population. So some associations with certainly some strong associations and then some other systemic associations. So again, if anybody's, this is like putting bells off in anyone's head of some sort of relationship, please let me know. And then again, just looking at some of the proteomics and genomics in keratoconus. This is just some of the different findings. So the keratoconus, those are gags in the cornea. Lots of different, a lot of these cathepsins, proteases, those are going to break down collagen, so reduce the collagen in the cornea, which is thought to be one of the primary causes. And so this is a really good, and this is the really it here. This is a good recent review paper, actually a few years ago now, looking at kind of the relationship between a lot of the different studied proteins and mRNA and the gene expression pathways and all the upregulated and downregulated proteins and genetic pathways. So there's over 30 different genes that have been found to be associated with keratoconus. So we're hoping that in the future, we do a lot of, we want to really dive into how the protein and genomic expression and a wide array of these cytokines and proteases is interacting together to create the environment that precipitates keratoconus. And so this is just some of the references that weren't listed throughout. And then again, acknowledgments to Dr. Lima and Dr. Assad, who are here today, who work in the lab. And there's several other people. And hopefully my list will grow longer and longer and take up the whole page like you guys by the next time I'm around. So thank you very much.
Thank you, Maria. You know, if you need tear samples, I can volunteer my wife. She cries when she watches K-drama. She cries when she watches The Walking Dead and someone dies. She even cries in Bridgerton, so, you know, I'll send her your way. Any questions for Maria Walker, Dr. Maria Walker? Actually, I do have a real question. With keratoconus, are you seeing any correlation with the quality of the contact lens? Someone who does like hydrogel versus silicone, you know, based on the materials of contact lenses. I mean, half of this group wears different types of contact lenses. So, what do you see in terms of inflammatory markers affiliated with the contact lens material?
Yeah, so I'll answer that a little bit more broadly than just keratoconus, because in keratoconus and some of these diseases, you're wearing a rigid plastic, which is going to be a lot less impermeable or permeable to things like proteins, lipids that could excite the inflammatory response. So people in this room, if you're wearing contact lenses, it's either a straight hydrogel material or it's a silicone hydrogel material. If it's a daily disposable, it's probably hydrogel. That's the best kind of option of a contact lens, because whatever gets stuck on that lens during the day is going to be thrown away and disposed of at night. But whenever you wear a lens, proteins, all those thousands of proteins, lipids, they glob and they stick onto the lenses. And so depending on what your cleaning regimen is and how good you're actually at doing it, and what your just natural inflammatory state of the eye is, I would say probably if you're in a Sci-Hi versus just a hydrogel, or regular hydrogel, the silicone is actually very lipophilic, so it will you will get more lipid deposits if it's a silicone hydrogel lens. Now, it's not that simple. The companies are very good at compensating for the weaknesses, and that's where the daily lens is great, because you just throw away everything on the lens. But if you have a reusable lens, you get proteins that will stick to the lens. They don't survive, so they break down, no longer recognized by the immune system, so you do get an inflammatory response, similar to what an allergic response would be.
Yeah, when I was covering Luminex assays back in New York, I worked with the Mount Sinai group that were working with dry eyes. It was, I think, Dr. Yiwei, Dr. Yiwei, and so he looked at interferon gamma, IL-2, TNF-alpha, and IL-10 in using a high-sensitivity inflammatory cytokine kits. And one of the findings they found is that, obviously it's common sense, but in dry eyes they found a stronger inflammatory expression. So one of the things that they were trying to work at next was those with contact lenses, those who still suffer from dry eyes, and how contact lenses actually relieves that to a certain degree. But I'm not an expert like you are, but I was just, that was just a little comment. Any questions for Dr. Maria Walker?
Thank you, guys.
Okay, thank you. Appreciate it. I'd like to introduce our next speaker. Came all the way from Minnesota, where the weather is not so great there. Dr. Jair Netto, he's a senior research technologist at Mayo Clinic. He's originally from Rio de Janeiro, Brazil, where I don't know if you wrote this, like you specialize in samba, you specialize in soccer and churrasco, or you meant Brazil specializes in that, but you can explain. In college, you majored in industrial pharmacy and Federal Fluminescence University in Rio. You had your master's in pharmacology and medicinal chemistry, where you study prevention of muscle damage induced by snake envenomation. So yes, we did have a conversation that you do fight snakes on the side. You worked with heparin, pre and post-treatment, EDL muscle, so fast muscle, slow, with twitch differences of response. Did a PhD in pharmacy and emphasis in biochemistry, enzymology, and drug development, whereas PhD work focused on glucose metabolism and NAD metabolism in cancer, diabetes, and SERT activators. Jair also did his postdoc at the Mayo Clinic Aging Center, focusing on cancer metabolism and NAD metabolism in aging and age-related diseases. He is currently working with Dr. Kirkland and Dr. I apologize, I'm not saying the name right, Keratoconus, at the Mayo Clinic Aging Center under the facility of Geroscience Analysis, which is an analytical facility focusing on biomarker research for biological aging. The title of his talk is Aging and Senescence, the Internal Flame of Inflammation. So I'd like to bring Dr. Netto on the stage. You okay with the microphone?
Yes.
Okay.
Totally fine.
A laser pointer and then left and right for changers.
Okay. So, can you guys hear me okay? Good afternoon, everyone. As Bob has just said, my name is Jair. I work with Dr. Kirkland and Dr. Ceconia. I initiated my career mostly in the basic and preclinical research, and around 2 years ago, I switched to more of the clinical research and clinical position analysis. And my postdoc focus was more on aging and a little bit of cancer. And I'll be talking a little bit about the aging and senescence and how these two could be related. So it's common sense that nobody dies of good health, right? We all die from a disease. And the diseases are super, can you still hear me? The diseases are super heterogeneous, like many tissues are gonna experience the decline. Aging is a decline of function. You're gonna see the change in the structure, and yet they are so different, you can still see that there's one thing that is common that links them all. They are more abundant in elderly people than in young. You're not gonna find commonly, unless there's like a secondary cause, a cardiovascular disease. And, oh, thank you.
Want that, it'll help you.
Yes, let me see if I... Okay, oh, better. So now I can see my slides here. So I was saying, it's extremely rare to see that in younger people, and yet it's very common to see in elderly people. And at some point, at the middle point of the lifetime curve, of the lifespan curve, you're gonna see those diseases going up exponentially. So everybody that studies aging believes that it's not just a coincidence. We believe that there are a biological mechanism behind the appearances of this disease related to aging. And today I'll be talking about one of these mechanisms and about the body of evidence that has been put together in preclinical and a little bit in clinical recently of how this process look like. So one of the processes that is involved is cellular senescence, and senescence is a cell fate. So a cell can go quiescent, like it's gonna stop the growth, but it's going to act as like a normal tissue cells. So under some stresses, another fate, and we are not sure about like how that happens in vivo, how the cells make the decision of going senescent. It's a state of growth arrest. There are some researches, it's very rare occasions that these cells can go back in the cycle again and maybe become a cancer. But this is like a fairly stable way of arrest. And the second point is, besides the cycle arrest, you have also the production of SASP, and SASP are inflammatory cytokines and other chemokines and other signaling molecules to attract inflammation. And the third characteristic of this cell is that it's also resistant to apoptosis. So these three basic characteristics of these cells is that it's gonna be resistant to apoptosis, it's gonna be highly inflammatory, and it's gonna have a growth arrest. This growth arrest is gonna be caused, among others, by expression of proteins that's gonna control the cell cycle. Most common ones we're gonna see around is P16, P21. Those are gonna make the cell stop growing and stop proliferating. And but like some cases, like you have P16 positive and P21 negative, but still senescence. That's not gonna happen all the time, all of them, and we're learning this from the genetic models. But those are like fairly markers that you're gonna see around. Also, another important marker, the lysosome of these cells is gonna start expressing a protein called beta-galactosidase that is gonna be able to convert some reagents into a blue dye in a different pH. Lysosomes are usually super acid, and this enzyme can function in physiological pH of like 7. So we can drive senescence in vitro in many ways. We can cause a DNA damage, and that, for example, we do an irradiation, and after 6 weeks, these cells are going to start becoming, it's going to be fully senescent along these 6 weeks. Also, the DNA damage can come from in vitro cultivation with like serial replication. You have the Hayflick number, around like 50 doubling times. The cell is going to become senescent because it's going to lose the telomere, and it's going to evoke DDR, DNA damage response, which are like any kappa beta responses, and you're going to cause like mitochondrial dysfunction. So this cell is going to acquire this phenotype of being highly inflammatory. And one of the things that happen with aging is that the tissue is going to start expressing, one, change in the matrix, and second, is going to show a low-grade inflammation that is sterile. You cannot find biological aging that is triggering, like an external agent that would be triggering this low-grade inflammation that is associated with aging. That's so profound that like an experienced pathologist can differentiate from a tissue from a young and from older people, and also because of fibrosis, but also because of these inflammations. So we have other causes that can lead to senescence. We have like oncogenes that can elicit senescence here, and this is one of the models for studying in vitro too. And all this, like the SASP from the cells can make other cells also senescent. And also a recent, very impactful are the DAMPs, the damage associated molecular patterns. So long story short, for example, in our lab, we show that like COVID spike protein can make some cells senescent through TLR3 receptors, for example. So all this is going to be making the cell senescence. All these can be involved. In vivo, there are not like any written stone mechanisms, but like a lot of these can contribute. So, as I spoke, the effects of this, many other aging mechanisms is to ultimately is gonna show some aging phenotypes, which are like loss of resilience. It's known that like elderly people, they can recover worse from a pneumonia than a young people. For example, dementia, atherosclerosis, and I want also that to make clear today that cellular senescence is not the only mechanism. It's one of the mechanisms that's going to drive the phenotype in the cell and the phenotype in the organism. So one of the clinical trials we had in the lab was we have aged like donors for kidneys, and we had like young individuals and old individuals, and we would take the fat from these people and analyze markers of senescence. And senescence is not specific, it doesn't have any specific markers, so you have to have multiple markers to characterize it. So here, for example, we used the proliferating markers with Ki67 marker, and we also use this protein that's going to mark DNA damage. So these two markers, you're going to realize that you have much more of them in the elderly population than in the young population. Also, a recent paper from 2021 from this group in France, they show that like obese patients, they have more, and that's the beta-gal, the enzyme I spoke before, that makes it to turn into blue, and they had like more more expression of this enzyme in the fat from elderly people, for obese people than non-obese, and also in different types of fat. And from that same patients from before, I don't know if it's possible to see here, but like all these blue areas are the areas where you have the beta-gal positive. And those are related to the BMI. So the BMI low, we didn't have much of the expression of the beta-gal positive cells. And in the beta-gal, in the BMI higher, like 32, we did have more beta-gal. So that shows that also obesity and other diseases, like those are pre-diabetic patients, you can also induce an accelerated aging phenotype. So senescent cells, they can cause diseases, inflammatory diseases, and diseases that come with aging. And one of the ways we showed this in the lab was we did an, they call autologous xenograft. So we took like the cartilage from the ear, isolated the fibroblasts, split in two. One we made senescence by radiation. The other one we just left as it is. And we also characterized these cells to make sure they were senescence and non-senescence. So how did we characterize that? We did the assay beta-gal to make sure they were senescence, the beta-gal positive. We assessed the mRNA levels for P16 and P21 to characterize the growth arrest. And we also assessed the protein level in the supernatant of SAS factors, which we use the multiplex system. And we can see that like there is this group of factors that was higher in the senescent cells. So these senescent cells was transplanted to the knee of mice. So the control group received the normal cells, and the other group that received senescent cells, we can see the difference, what resembles what's an arthritis, Osteoarthritis, like we have like some mild osteoporosis around here, like some calcifications, showing that these cells can cause disease. and are related to this. So in another approach that our group published at Nature Medicine back in 2018, the senescent cells from pre-adipocytes from humans was injected in mouse, and we followed the longevity and the capacity and the, what they call like frailty index of these mice. So very small amount of senescent cells were able to induce a phenotype that resembles aging. So we had like a loss of grip strength. There was like this hanging endurance. The mouse was like less able to keep attached to this wire. And the maximum speed that that mouse could do on a treadmill was reduced. And we can also see that there was a latency period, but then these mice start dying in a faster rate and before the other ones. So that shows that also senescence, a small amount of senescent cells can cause an impact in mortality and also in the phenotype. Another interesting things was after 6 months, what was like the sacrifice, the euthanasia here, we could see that the normal cells, we didn't have the senescent cells, the luciferase. Those cells are luciferase positive, so we could track. They were gone, but yet the other cells from the body became senescent, showing that if you have senescent cells in your body in one spot, which was in this case the peritoneal space, the senescence can spread and can cause senescence in other parts of the body, in other tissues. So it's an interconnected dynamic of the biology. So all this led to the discovery of the drugs called Senolytic. And the way they did it, as we spoke, the cells are resistant to apoptosis. And it was very hard to have these cells eliminated from the body. But like a couple of work before, they found out in 2016 was word from Judy Campisi from Buck Institute. She showed like this inflammatory phenotype of these cells releasing these factors. And Ned Sharp, he's Sharpless, he's from NIH now. He showed that some intervention can delay or eliminate these cells too. So the elimination of these cells or the prevention of the growth of these cells in the body are preventable. So the group started doing like what resembles an siRNA arrest through these apoptotic pathways and tracked which one could kill or reduce the viability of these cells. And what they found was like not one pathway, but those pathways were interconnected. And that's how they got to the cocktail that is called D and Q. It's like the dasatinib and quercetin, that they can actually interact and block multiple pathways of this senescence pathways and apoptosis resistant pathway, and made the cells, the senescent cells, less resistant to its own SASP, to its own inflammatory cytokine, so they could be finally like killed by their own SASP. So that's how. Another thing like that is from the field, you don't have like one senolytic that's going to work for all cells, because different senescent cells from different tissues, they can behave differently. They have different SASP, they have different types of phenotypes. So one of the limitations for now is like we don't have one drug that's going to be working for all types of senescent cells. So one very elegant way to prove that was the first clinical trial of the lab with tissue explant. Those are kidney donors, healthy kidney donors. And what they did, they took the biopsy, they split in two, and they incubate with D and Q to try to assess if they were killing senescent cells in vivo or ex vivo, but like in human samples. And what they found was a reduction in assay beta-gal positive assay. So the cells, the beta-gal positive cells were less abundant. They found less markers of DNA damage, like the excuse me, like the telomeres and the H2AX, which are like markers of DNA damage. So that was a characterization that these cells are being eliminated in this tissue. So each individual was like kind of the control of itself. And with that, we also analyzed the tissue itself to see the expression of some of the genes. So we analyzed the level of the mRNA, but also a cytokine array to make sure we were having the same effect that we have in the mRNA we would have in the supernatant or in the conditioned media from these two explants. So there was like a highly significant change in IL-6, IL-8, MCP-1, and PI-1, and those cytokines are very common in a huge amount of different senescent cells. So even though we have different SASP from different cells, there is a core body of proteins that is always going to be there, and those are one of those. So IL-6, IL-8, MCP-1 are going to be there for sure. And so this other study from this group from French, and it's independent of our lab, and like you're going to see that in many other publications too. They divided these people in 3 types of assay beta-gal. So they used the beta-gal assay to differentiate, to put in 3 groups. And they took the high beta-gal expressing or high beta-gal positive tissue, split in 2 and treated with D and Q. And what they saw there, one, there was a reduction of beta-galactosidase positive cells, which express here in the tissue explants, but they also were able to see in the supernatant these proteins that we referred before, IL-6, MCP-1, and PI-1. We didn't access IGF-BP3, but we do that in the clinical panel now, and it's interesting to see how this result's gonna being reproduced like in vivo, in vitro, and like preclinical and clinically. So that also led to the first human clinical trial that the lab did, was a study with like kidney disease, diabetic patients with kidney disease with the same drug, the cocktail of D and Q, dasatinib and quercetin. And we made a full characterization of the senescent cells in fat and in skin. So when you see here in the skin layer, that is the layer of cells that are proliferating. So we count the number of P16 positive in the cell in the membrane layer here, and also the P21. And what we found out is in patients treated with D and Q, 14 days after, there was this significant reduction in the number of P16 cells and P21 cells. So, and also in fat, we saw a reduction in beta-gal positive. And one of the things that is clear at this point, as we don't have a specific marker for senescent cells, we need a system that is able to measure multiple targets at the same time. And that target is important, that correlates with some phenotype, for example, the frailty that was measured here. So what we actually found was a group of cytokines that correlated with the reduction of P16, of P21, and with the phenotypes too. So the idea for now, it's beyond to try to analyze just younger nodes, since like aging is such a diverse system. The idea now is to find composite score that's going to respond to senolytic treatment and it's going to correlate with a healthy aging. So the idea is more to make a healthy aging over live longer. If you think about like C. elegans, it's one of the models to study aging in preclinical. The life extension was tenfold for that small. When you go to mice, it's like 5 to 20%. 20% life extension is like something that you have to celebrate. Usually it's 5, 10% at life extension. So the idea is not to make us live forever, but make us to have more, to be healthier for a longer time. And that's going to have a huge impact in many areas, from economics, social security, employment and everything, but mainly in the quality of life of people, because some of those diseases is not going to kill you, but it's going to have a huge impact in the quality of life. For example, macular degeneration, carpal tunnel, osteoarthritis. It's not going to kill you, but it's going to reduce a lot the quality of your life. So our idea is, if we can aim at these mechanisms of aging, and it's not like just related to senescence here, we have multiple interventions. The idea is that if we can reduce the age-related disease. So that's the next step. So based on that, a group of gerontologists created the Translational Neuroscience Network, short-term TGN, which are a group of people that are creating a way to standardize and create an infrastructure for aging research in humans. It's very hard and challenging to study aging research in humans because of the big longevity. So we're trying to find biomarkers of healthy aging, and especially the ones that's going to respond to intervention, like exercise, caloric restriction, and also, of course, drugs. We have like the NAD boosters. So the idea is to have this TGN as like a core for infrastructure, and also the FGA, which is the Facility for Neuroscience Analysis, which I'm under, is the idea to develop methods that we could measure these biomarkers. So it's more of being a research use type than like being a clinical lab. The idea is to have like a big array and find whatever is going to be interesting, of course, with a robust assay, an assay that's going to be reproducible. And then when we have a final composite score that is going to correlate, we're going to go to the next step. We then, like for the biomarkers of aging, we target multiple areas. For example, the secretory phenotype, we can do that in vitro, ex vivo, and also in biological fluids. So, for example, we have blood, of course, but we also do urine. We are developing saliva, because those are also valuable matrix, because they're going to show you the unbound proteins. And some proteins, when you measure in plasma, they're going to be a mix of bound and unbound. And mostly those tissues are going to show you unbound. So here's like a point of very importance to us for the composite score, and also the markers of senescence to show the pharmacodynamic, the proof of concept that the Senolytic is going to be working in different tissues, in different areas. So we do a lot of flow and microscopy. Also DNA damage markers, telomere markers, and morphology is also a very important part for us. And so those are the clinical trials that we have now. Some of them are ended. So the ones that are blue are with senolytics. The ones that are not blue are without senolytic drugs. And we have different types of study. Those are, for example, entolimod is one for vaccine. It's going to see resilience. Elderly people, they have a worse response to vaccine. So there is a drug called entolimod, and they are seeing if that can increase and make better the response in vaccinated elderly people. There is a study with metformin. There's like a continuation of the TAME study that's going to also be started soon. We have like studies with Alzheimer. So different tissues, different areas, and the final idea is to improve the life quality. And last but not least, our next interest is also the Axiom project. Is the one I've been working hard recently to get samples or people that are going to be sent to space, and also some cell plates. We're going to analyze before and after and see if the radiation in space close to Earth is going to affect any of the proliferation markers and markers of senescent cells in vivo of people and cells, human cells, that are going to be shipped to space. So this is our group, Dr. Kirkland, James Kirkland, Dr. Ciconia, and this is like the two labs together, FGA and Dr. Kirkland. And thank you for your attention. Hope we could have a message here.
I don't know about you, but I'm going to go to Dr. Simmons, get a prescription of Senolytics, and I'm going to go to my local CVS right now. Any questions for Dr. Jair-Netto? I have a quick question for you, because this relates to what I used to do in graduate school at MD Anderson. I used to work with aging in the prostate, and one of the things that I worked with that you're doing right now is that when you treat certain tissues or certain cells or epithelial cells with Senolytics, you see P16 going down, you're seeing maybe possibly hTERT expression, I don't know, maybe going up. Have you thought about maybe from a Luminex perspective, homogenizing those cells or collecting cell culture soup? So let's say osteoarthritis, to look at OPN reversal, because if you do reverse aging, you would assume that a lot of the bone metastasis markers may be going on decline. If you were doing with obesity, whether or not leptin and adiponectin, so forth, would actually go reverse. So have you looked at some of that where you can actually, from a multiplex perspective, collect either cell culture soup or homogenate those? Because senescent cells, it's very difficult. You know, you can't homogenize senescent cells because there's very few in the population. So I was just wondering, from a future direction, or is that one of your plans that you may do?
Yeah, so one of the things we do in the basic is actually we do that. We used to do ELISAs, and thank you for saving my time. We do because some of the proteins that are there is not going to be secreted. So we do have part of it that's going to be like homogenizing and looking. And also one of the things you mentioned about cancer, senescence is supposed to be one biological mechanism to save the cells from becoming a cancer cell. So it's going to stop growth and arrest, right, so it doesn't become a cancer cell. So the phenotype and the expression of proteins are going to be different there.
Right. Any questions for Dr. Netto? Okay, so we are on time. We will, our next talk is at 2:45, so let's take a quick 5-minute bathroom break. And then, yes, if you can just come back in literally 2:42.